We started from the molecule glucose or other sugars, and we went down what pathway with that sugar? Glycolysis, right? And we made some energy, right? We made some energy molecules, and we left those in the cytosol. Then we transported some of those energy molecules in the form of pyruvate into the mitochondria, the powerhouse of the cell. And we did some more reactions. What pathways did we invoke within the citric acid cycle? What were the pathways? Well, which one? The what? We had to create acetyl-CoA. It's a one-step cycle, the pyruvate dehydrogenase complex, to make acetyl-CoA from pyruvate. And then that acetyl-CoA went into what pathway? Citric acid cycle. And throughout the whole citric acid cycle, for every pyruvate that you brought in the pyruvate dehydrogenase complex, one CO2 flew off as waste. And you went through the citric acid cycle, and 2 CO2s flew off in that process. And you created lots of energy molecules. Okay, most of those were NADH, but there was also an FADH2 produced, and one GTP that we call ATP. Okay, and we had to go through the citric acid cycle twice, right, to get all of the carbons from glucose completely oxidized. And so we have lots of energy molecules inside the mitochondria from which we can extract some energy. Now, what we are going to do in the electron transport chain, or oxidative phosphorylation, is look at those electrons and flow them to oxygen, if we can, and hopefully we do. And we're going to convert all of that energy into a proton gradient. And that proton gradient will be the amount of energy that the process creates. And then in a separate process, we're going to use the energy of the proton gradient to create the molecule potential energy that is an ATP. All right? And so we have complexes by which, or proteins by which, the electrons are shuttled through, and we have some carriers that we introduced last time. All right, and we looked at some differences. So let's start off with complex 1, and if we start off with complex 1, we need to know this. We are dealing with the electron carriers of NAD itself, okay, in complex 1. And so NAD, and then we always carry around an a proton with that, will give its 2 electrons into the protein called complex 1. Now, we've got a pretty good crystal structure of the protein portion that actually resides inside the matrix. It's the soluble portion, and that soluble portion can be cut off and it can crystallize pretty well, and you can see the structure that we have here. Now, what's important about this structure, when you do crystal structures, the larger the atom, the more scattering, the more we actually see it. And so we see very well the iron molecules, the iron atoms that are in these chains that kind of have a pathway here. All right? And you can see them there in this picture. Now, remember what we're doing. We're making electrons jump. And so NADH carries 2 electrons. We're going to give 2 electrons, and they'll be accepted first in an FMN organic structure. That's the same organic structure as FAD, so it can hold 2 electrons. And then we've got iron centers, which the electrons will jump place to place to place. Now, what you can see them actually in the structure. They're embedded, and it's almost like a continuous metal wire in which the electrons flow. What is interesting about these is that they are very close to each other, so the electrons do not actually jump very far in distance from place to place to place, because they're not actually, it's not a true metal wire because it's not completely touching, but it goes from one center to the next. Remember, every single transfer from one entity to the other, we lose a little bit of energy, because we're not extracting all of the energy that we could possibly, okay, because we calculated only 34% of it went into ATP of the potential energy of the electrons actually getting to oxygen. Now, we don't have a really good sense of what the players are in this area, because in the portion that's very hydrophobic, they don't tend to like to crystallize very well, and so we don't have a good structure, so we don't know exactly those places. We have some ideas, but we don't have as good a measure as we have here. And so we are throwing all these electrons through this process. Now, here is the important part, okay? This Q right here has no electrons, and this Q right there has electrons, okay? But what is different between those two things that are circled is that those Qs are mobile, meaning that they are able to float into the membrane, and they float up and bind to the side of the protein. They're not inside, but they bind to the side in a pocket where they pick up the electrons, and then they can diffuse back out. All of the other things, the way that we write it, it's in the middle of the protein. So this is mobile and that is mobile. They move in and out and bind to a pocket to catch those particular electrons. As a consequence of all of the movement, we get, and this is the biggie, 4 protons pumped across. Okay? This is where the energy is converted. Okay? The last step, it says here, the QH2 floats out and into the membrane, and I create a very large concentration of QH2 in the membrane. That means I have a lot of energy floating in the membrane, and that energy can then travel to the next entity. Okay? So for every 2 electrons that flow through, 4 protons are pumped. Now, complex 2 is a little different. Remember, this is where we are going to allow the electrons to come in, okay? And so this slide is kind of messy, okay? So let me identify a few things. This right here is complex 1. We just covered that, so that's not what we're looking at. What we are looking at originally is this, the FADH2 that comes from the citric acid cycle. Now, really, what we discovered was that complex 2 was the citric acid cycle's enzyme that produced the FADH2. And so it is the enzyme that produces it, and what happens is the FADH2 is accepted, or the electrons are accepted in the FADH2 that is bound to that enzyme, and then immediately it gives it to its iron center, and then it immediately gives it to Q. And I don't like the slide because when it accepts the electron, it's QH2. Okay, so it gives it to Q, and it becomes QH2. Okay, that is the normal process for the FADH2 that is produced in the citric acid cycle. Okay, it never actually leaves that complex. It's actually bound in part of that protein, and it's part of that enzyme. Now, there are a couple other places. One is here, and one is here. Let's talk about those 2 places. Okay. Let's talk about this first, fatty acid oxidation. Okay, we're going to learn that in the process of fatty acid oxidation, as we break down fats, we're going to produce an FADH2 in the process. And when I do that, the electrons in the step that produces FADH2 will go into Q to create QH2. So the energy from fats, if ever it is in if ever it's in FAD, goes into QH2 at this point. Okay, that's important. Now, this one right here is hugely important. Okay. Now, we produced a lot of energy molecules, both in glycolysis and in citric acid cycle. In glycolysis, we ended up with 3 energy molecules, pyruvate, okay? And we know that that got put into the matrix. It was still potential energy at that time, and we completely degraded that. The other 2 entities was ATP, which is the energy molecule, and we put ATP in the cytosol where it should be because that's where it's going to be used, so that's great. But we also made an NADH, okay? We actually made 2 of those. The NADH that's in the cytosol is in the wrong place, okay? And so one way that NADH from the cytosol that came from glycolysis that can get into the electron transport chain is to go through this enzyme right here called the glycerol 3-phosphate dehydrogenase. The electrons from NADH will be converted into glycerol 3-phosphate, and then that glycerol 3-phosphate will take the electrons and put it into FAD, and to become FADH2, and then it gets put in to become QH2. Okay? And so the NADH from the cytosol gets converted into FADH2 at this step. That's a possibility, one possibility, and I get QH2 into the electron transport chain. What do you not see happening in complex 2 at all? Which happened in complex 1? Yes, no protons go from the inside to the outside. What we have done is put the electrons into QH2 so it can complete the cycle, but no protons are pumped across. Remember, the energy is converted into that differential or that proton gradient, and here I have energy flowing, but I get no net energy to make ATP. Okay? Anytime the electrons are carried by FADH2, I am going to get less ATP out of the system than if I carried it through NADH only. Okay? All right. Now, I've got, at this point in time, a lot of QH2 and high concentration, because I've been making it from complex 1 and for complex 2. I get lots of QH2 just roaming around in the membrane itself. All right? The next place that QH2 will give its electrons is to complex 3. Okay? It's got, this book names it ubiquinone, which is the name for Q, cytochrome C, octoreductase. So this is nice. It tells you it comes from QH2, gives the name of QH2, and it gives it to the protein cytochrome C. All right? Here is the problem that we have to solve. QH2 carries 2 electrons. That's all well and good. Cytochrome C carries 1. That's a problem. How in the world do I have a 2-electron carrier come in, give 2 electrons, and it go to something that only carries 1 electron? Okay? And so we're going to have a series of events that occur that allow those electrons to flow through. We got a pretty good idea of the structure, crystal structure of this. There are lots of components. And so the important parts of this, okay? It's kind of little, all right? But there is a cytochrome C that is dotted up there. That means it can come in and go out. So it's mobile. It binds to the protein, and then it can float away. And that's where the electrons are going to float away. There is a heme that is permanently bound in the complex. Okay, and that's equivalent to the cytochrome C heme. There are a couple other hemes that are part of this. There is a heme B version, that's the L, and a B heme version, that is the H. So a heme B high and a heme B low site. And then we're going to have some Q binding places. All right. Important, as 2 electrons move through this, we're going to have 4 protons pushed across the membrane. All right. When you see a slide like this, what do you feel? Sad. Yes, very, very sad. Okay. Now, we're showing a cyclic process, and putting that on a piece of paper is really, really hard. So let's start with the first step. Okay? First step is right here. Number 1. A QH2 that has 2 electrons that's right happy comes in and binds. And so it is bound in step number 2. All right? And it binds into a pocket. Now, at this point in time, what's going to happen is 2 things. All right? One thing is, let's do, stop. Okay. One thing is this. It's going to take one of the electrons and go here. Let's call this 3A. Okay, it's an iron center. Then the electron's going to hop to the cytochrome C that is bound. Let's call this 3B. And then it's going to give its electron to the cytochrome C that can float away, and we'll call this 3C. That is 1 electron. Okay. Now, at the same time, 1 electron is going to go down here. Okay. And I'm going to call this one A3. Electron goes to the heme low site. Then an electron goes to the heme high site. We're going to call this B3. Then I'm going to put the electron onto a different Q that binds to the protein. And that is C3. So 1 electron goes up, and cytochrome C floats away. And 1 of the electrons goes down, and it sits on this Q. Now, here's the confusing part. See that arrow? And you see this dotted line? What really happens is the Q becomes this. It holds 1 electron. It has a negative charge. It doesn't move. It's just sitting right there. It converts from Q, that is neutral, to this, a Q that has 1 electron. Okay, but it looks like it moves. It's not moved. It's just kind of sitting there holding the extra electron. That Q that is now bound to the side of this protein that has a negative charge can't move. It's stuck there. All right. A second QH2 comes in. All right. Now, what's different is this second one has this, a Q that has the negative charge and an electron. So before, it just had a Q. And now I have a new cytochrome C that can carry another electron. So this Q comes in, QH2. It will give 1 electron up here. This floats away. And it gives 1 electron down here. And it has 2 electrons. And it becomes QH2 by picking up protons. And this QH2 can float back into the pool of QH2s. So what happens is, net, I have 1, 2 coming in, 2 QH2s coming in, and 1 QH2 regenerated. I also have now 1 cytochrome C taking 1 electron away and 2 cytochrome Cs taking the second electron away. So I bring in 4 electrons, 2 and then 4. I have 2 electrons that go to the next step, and 2 electrons are regenerated into a QH2, which becomes the pool that was there. All right. This is how we can cycle in and bring in a 2 electron carrier, send only 1 electron to be carried, 1 electron to be held for a moment, then we send in another, 1 gets sent to the other, and 1 gets regenerated as QH2. All right. Complex 4 is interesting. Okay, and complex 4, the stoichiometry changes a little bit. Up to this point in time, we've been looking at 2 electrons flowing through. So we either had the 2 electrons from NADH or we had the 2 electrons from FADH2. Here, what we are bringing in is oxygen, and we're going to produce water. Okay, but the stoichiometry changes because for the transformation from oxygen to water, I have to have 4 electrons to actually do that reaction. Okay? And so, what in the world is going to happen here? Well, we're going to bring in oxygen and we're going to trap it between 2 metals. Okay, we're going to put it in a really, really bad place. We're going to put it in a place that it's going to get electrocuted. Okay, we set it right between 2 metals. Now, but that's what we want. We want electrons to flow into oxygen so that that's where all the electrons will end up. And oxygens are fine. They'll take electrons, and when they get electrons, they become negative. And when they're negative, they pick up protons. That's where we get the water from. And so the process happens this way. We've got 2 special metal centers. One is iron and one is copper. In their oxidative states, originally in the 3+ and in the 2+, so 3+ iron and 2+ copper, oxygen can't fit. Okay? In terms of the number of coordinations that those atoms can actually make, there's no place for oxygen to fit in there. But we throw in 1 electron. This electron here comes from cytochrome C. Okay? And the copper accepts the electron. And then we throw another electron into the system, again coming from cytochrome C, and we get the 2+ iron and the 1+ copper. And it creates just enough coordination that oxygen binds into it, but not perfectly. Do you see how it's bent in there? We've seen oxygen bind to and coordinate to a metal in a bent way before, and that's in hemoglobin. It's a weaker bond because if you want a good bond, you want the overlap of electrons to be directly in line, so you have a nice overlap to create sigma bonds. If you're coming in at an angle, you can't overlap as well between the 2 atoms. So it creates a weaker bond to oxygen. We do not want to bond strongly to oxygens, because if we do, we would lock it up and we would make that oxygen stick in there, and it would never be able to be released, never be able to be accepting these electrons. By having this weakly bound, it allows us to do the next process. All right, I've got oxygens in there. That's great. Now, this is what happens. Copper gives an electron in. Iron gives an electron in. And now, it is not oxygen. It is an oxygen that has a -2 charge. All right? That -2 charge on those 2 oxygens allows it to be bound between the copper 3, and the copper 2. But I'm not done. I throw in one more electron. Where does that electron come from? Cytochrome C. And 2 protons. And what I get is one water that is, one of the lone pairs is coordinated to copper. Okay? It shouldn't necessarily be a full bond. It should be a dotted line because it's just a coordination event. Okay, but I've created one of the waters, okay, that I want. And then I get this thing. Okay? Does that look nice? Does that look like something you've seen in general chemistry? No, this is a very, very unstable oxidative state. We haven't really seen iron in a +4 state, double bonded to an oxygen that has a -2 state. All right? Very, very not happy. So what do? We throw in one more electron just because. Okay? And when we throw in that extra electron and another pair of protons, we form the second water, and both waters leave to produce the beginning state of the complex 4. Now, we show you this in terms of 4 electron flows because we wanted to get to the ultimate product of water. All right? Now, if you actually looked on that slide, it said 4 protons were pushed out, right? That is in relationship or stoichiometry of 4 electron flows, but we've been working everything up to this point as 2 electron flows. So if 4 electron flows gets 4 electrons, 2 electron flows gets 2 protons. Okay? And so going back to stoichiometry that we were counting as 2 electron movements, we get 2 protons for 2 electrons. And so for NADH, we get 4 protons across the membrane for complex 1, and in complex 3 I get 4 protons across, and in complex 4 I get 2 protons across. And in complex, or for FADH2, complex 2 doesn't get any protons across, but I do get them into QH2, and I can do complex 3 and 4, and I get 6 across. Okay, what is important in this? You need to know how many are pumped out by each of the complexes in a stoichiometry of 2 electrons. Okay. And how are they different? And so if I look at that with FAD, none there, but FAD does get that one, and FAD does get that one. Okay. I want to go back and correct myself real quickly. Remember, we need a proton gradient, and I said this right here, if I take those protons and I put them into water, that increases the proton gradient. I incorrectly told you last time. This process here, there's a proton to get here. I actually reduce the number of protons, and that actually does increase the gradient. Whereas I said last time in the lecture that it decreased, it actually does increase the gradient. Although no protons are pumped across, it has increased it. All right. Now, what I've also done is I've recreated NAD plus, and I've recreated FAD. Those can go back to the citric acid cycle and be reused again. And so they can take the electrons from the citric acid cycle, bring them to this, and go back and forth, back and forth, back and forth. Okay? All right. We have cytochrome C carrying electrons right here in water. Here in complex 1, all in hydrophobic conditions. Q, all in hydrophobic conditions. Complex 2, hydrophobic. Complex 3 and 4, hydrophobic. All of the rest of the players carry it in hydrophobic areas, but cytochrome C puts the electrons into an aqueous layer that is different. All right. So NADH got 10 protons. FADH2 has 6 protons. Let's do some calculations. Oh, not yet. Oh, stop. All right, I'm going to do some calculations just on this sheet of paper real quick, or on this slide. How many ATPs did I say NADH will produce? How many ATPs? Do you remember? Not just 2? It will create 2.5. Okay. How many from FADH2? 1.5. Okay, let's do some math. All right, I pump out 10 protons and I get 2.5 ATPs for NADH. How many protons per ATP? 4. Good. Even without your iPhone, you did it. I am very, very happy. Okay, so 10 protons divided by 2.5 ATPs for NAD plus is 4 protons for every ATP. Good. And over here we had 6 protons, 1.5 ATPs for FADH2. So what's the calculation? How many protons per ATP? 4. All right. So we know how many protons it takes to make 1 ATP. It's 4. Or so we think. Okay. All right. I have completed the electron transport chain. At this point in time, all potential energy has been used. The glucose has been converted all the way to CO2. There's no more potential energy in those carbons. I have them in the form of ATP in the cytosol from glycolysis. I have some ATP in the matrix from the citric acid cycle. And I have a bunch of protons outside the matrix and very few protons inside the matrix. That's where all the energy is. The next step is to work with the ATP synthase. Now, the ATP synthase is a protein that is an integral protein. It does go through the membrane. It has a couple of pieces to it. All right? There is a place in which protons can come back through. Okay? So it facilitates protons going from one side to the other. And this little piece right here is actually going to spin as the protons go through. There are 3 different proteins, okay, gamma, epsilon, and delta, that are attached to this place that spins. Okay, so you can see the rotor here, and you can kind of see the proteins right there. But that's not all. The protons actually flying through a portion through here, and it's going to go in at the top. It has to bind into this rotor part. It's going to rotate around, and it's going to come out the bottom over here. And you can see that these are connected. So the protons come in, they get transferred over here, they get moved around, and they come out the end. Okay? That's the stator. Has anybody ever heard of rotor and stator before? Okay. Rotor and stator comes from terminology in an electrical engine. Okay. These are very, very close to and in the same structural components as an electrical motor. And so we use those terminology. Then we have what's called the head piece. And it's 6 proteins that are alpha beta subunits. So I got 2 alpha beta, and then 2 alpha beta, and then 2 alpha beta for 3 different states. Okay? The head pieces are where the reaction is going to occur. All right? So, we have a place where electrons flow between this and this rotor. We know that these pieces are going to spin, and they're spinning inside the area between these 3 pairs of alpha beta And this delta, epsilon, and gamma, or delta, epsilon, and gamma proteins actually will touch these pairs. Okay, and they will influence the shape of these pairs of proteins. Okay, the place where electrons flow through is called the FO. That's not F0, but an FO. It is O because oligomyosin actually can inhibit or bind to that section and prevent the flow of protons. Myosin is a general term, a general drug term for an antibiotic. And so this one mechanism for one type of antibiotic is to stop the synthesis of ATP. That should kill something. If you can't make ATP, you can't live too well. The F1, just to be confusing, section is where the synthesis happens. And if I actually isolate the F1 portion and put it in a test tube, it actually works as a ATPase. What does ACE mean? It's an enzyme, and usually it's an enzyme that breaks things down. So an ATPase is an enzyme that breaks ATP down. But remember, the transition state of breaking and making are exactly the same. So I'm going to stabilize that transition state. Okay? All right. So this is the actual transition state. All right, ADP and PI, they'll come together. What actually happens is this pair of electrons will act as a nucleophile. It will attack the phosphate, and in the transition state, the electrons kick up. What that does, it creates this phosphate becomes this phosphate. And it's called a pentocovalent intermediate. Why is it called a pentocovalent intermediate? 5 bonds to the phosphate. What is the shape of an atom that has 5 bonds to it? What is the name of that shape? Trigonal bipyramidal. What is its hybridization? S, P, how many P's? 3, and a D. You have to have 5 places for electron pairs. You take your S, all 3 P's, you mix those together. I don't have places for 5, so I have to throw in a D. Okay? All right, that is the transition state. This is what is stabilized. Do you see this transition state? 1, 2, 3 negative charges. That's hugely negative. All right? To finish that transition state, the electrons on one of the pairs will fall down, and what will happen is the hydroxide will leave as water, and we get ATP. Although we could reverse that and break it down. ATP, have water come in, and create ADP and PI. Okay. Here is where crystallographers are just magic. Okay? They have a crystal structure that has a pair that has no ADP, PI, or anything. Okay? Here's a crystal structure that has ADP in the active site, and here's a crystal structure that has in these two an ATP bound. Okay. If I look at that and I don't have these outlines, that and that and that to me all look the same. But a crystallographer, and you look at the actual structure, this is totally a different shape than this one and this one. Okay? What we're going to call this shape right here, and what they call this shape right here that doesn't have anything bound to it, they're going to call it the open site. This shape that's a little bit more compact actually has ADP bound and a PI bound. This is called the loose site. And then this particular conformation that is the most compact actually has the ATP bound. Okay? And so these three different sites are slightly different. One is kind of open. This one is kind of closed a little bit. And this one is totally tight in terms of it's really close together, like pushing things together. And so I have three different things going on in these different pairs of the two alpha betas. Okay, here's an experiment that they set up. They actually put the head group and they bound it to a slide on a microscope. They also then put on a binding molecule that bound to this rotor portion, and then they put on an actin filament that had a fluorescence dye so they could see it. Okay. What they did was they took an electron microscope and they fed it some acid. Okay, not that kind of acid, but real acid. Okay. Before they added the acid to it, they saw the actin in one particular position. They added some acid, and what they found was there was a transition. And the angle between this orientation and this orientation right here was 120 degrees. And it stayed in that position for a little while until they added some acid and they saw a transition. And now it's pointed at about 120 degrees in another orientation. And then they added some acid and it transitioned back into the original orientation. So as protons flowed through, it changed or rotated itself around. Okay? Now what really happens here, as this rotates, what happens, remember there are three proteins on the inside. At one point in time, one of these proteins is interacting with one AB pair. Another protein that has a different shape reacts with these two pair, and the third protein that has a different shape react with these two pairs. Depending upon what orientation, those pairs will either be interacting with a delta, an epsilon, or a gamma protein. If you're in an epsilon protein, or a gamma protein, or a delta protein, you're going to have one of those three shapes, the open, the loose, or the tight. And as I move those proteins from pair to pair, it changes their structure. One goes from open to loose. One loose becomes tight. One tight becomes open as that combination of the AB interacts with one of those three proteins. And so what we see is up here in this portion is that in one orientation, the open had an ATP, but when it becomes open, the ATP flows away. Okay? In the loose, the ADP and PI are trapped together, but they're not pushed together. They're just trapped together. And when it's tight, the protein closes in on itself, and it pushes those two reactants together and stabilizes that transition state. And then protons flow, 3 of them. And what happens is this which was loose, this which was open, the ATP fell out, an ADP and PI fell in. Now it's in the loose state. This loose state is now in the tight state. I convert those two into ATP. And this tight state gets converted into the open state where ATP falls out and then ADP and PI can bind. 3 protons flow through. This that was the loose becomes the tight. This tight becomes the open. This open becomes the loose. 3 protons flow through. The loose becomes the tight. The tight becomes the open. And this pair that was the open became the loose. And so it just moves these around. All right. It takes 3 protons to rotate around to create 1 ATP. What did we calculate a few slides ago? 4. But we really know it only takes 3 to make that rotation occur. Okay? So where in the world is that lost proton? Okay, oh, here we go. This was the calculation. So we had 4 protons per 1 ATP, and we actually see that that's absolutely true. But only 3 protons have to flow through to make. So that means we have 1 lone proton that is missing. Where is it? Okay. Where is the ATP made that we just saw? What compartment? It's the headpiece, and then it's released into what compartment? The inside of the mitochondria. Where do we need the ATP? In the cytosol. And when ATP is used in the cytosol, what is produced in the cytosol? When you actually use ATP, what is left over in the cytosol? ADP. But I need ADP inside the matrix to do my synthesis, right? I got that in the wrong space. Okay. So what happens here is this. All right. We did make all of the ATP here. I do need it out here. And so what happens is, and they should have shown this in the same place. Okay, that protein and this protein is the same. All right. When ATP comes up and binds, it'll go through the channel, open up, and ATP will float away. And now it's open to this side. This has no affinity for ATP. This has a huge affinity for ATP. What does this now have an affinity for? The ADP. ADP will bind in and open back up to this side. This has no affinity for ADP, so it floats away. And so I keep the balance of ATP and ADP perfect. Every time an ATP goes out, an ADP has to come in. Okay? But to do this reaction, I have an ADP. I also need a PI. So I produced ADP and a PI in the cytosol. To get that in, here's the PI has to come in. For every 1 PI coming in, what do I have to have coming in with it? The proton, the lonely 1 proton. This is that 4th proton that is needed to make an ADP. 3 are used for the synthesis, and 1 is used for the transport of the PI, so I keep everything balanced. Okay, so it does take 4, 3 through the synthase, and 1 to make sure I have the reactant, the inorganic phosphate. Okay, the stoichiometry is important to know. You should know the stoichiometry. You should know where all the protons are used when you make ATP. Got it? Okay. All right. Now, let's complicate stoichiometry just a bit further. I've actually given you a preview to this. Okay. NADH, how many ATPs do we make from NADH? 2.5 star. In the matrix, I will always get 2.5. Okay. The NAD from glycolysis, though, 1 NADH can make 2.5 ATPs, or it can make 1.5 ATPs. Okay? It all depends upon how the electrons get through the mitochondrial membrane. Because NADH cannot be transported across. There's no protein to make it go through, and it's polar enough not to be able just to diffuse through. So there is an issue. there are two different pathways. One is called the glycerol 3-phosphate shuttle, and one is called the malate aspartate shuttle. Okay? I actually showed you a couple slides ago the entrance through the glycerol 3-phosphate shuttle. And this is what happens. Dihydroxyacetone phosphate is a glycotic intermediate, yes? Okay. What enzyme produces dihydroxyacetone phosphate? What enzyme of glycolysis produces that? It will also make a 3-phosphoglycerate, or 3-phosphoglyceraldehyde, sorry, 3-phosphoglyceraldehyde, and a, what is it? Yes, it was aldolase. Okay, so this is produced by aldolase. If by chance I have lots and lots of NADH, okay, that needs to get into the mitochondria, those electrons, one reaction that can occur is this. I react the dihydroxyacetone phosphate into glycerol 3-phosphate. Okay. This is not a glycotic intermediate, but this can be transported into the intermembrane space, and it can react with an enzyme that is glycerol 3-phosphate dehydrogenase that is in the mitochondria. In this, the electrons that were once in NADH get put into FADH2, and FADH2 puts it into QH2 without pumping out protons. Okay? Normally, the NADH inside the matrix goes to complex 1, pumps out 4 protons. I can use those protons to make an ATP, right? Here, the NADH up here, that's in the cytosol, if it goes through this pathway, I get electrons in, but I don't get all the protons pumped. Okay? And I have to do this twice because I have 2 of these that come from glycolysis. And so I can only get 1.5 rather than 2.5. And so for the 2 NADHs in glycolysis, if I use this pathway, I get 2 ATPs less than the maximum amount. Now, in the malate aspartate shuttle, it's a whole different reaction. All right, aspartate. What kind of molecule is aspartate? An amino acid, okay? It can react to create oxaloacetate. Oxaloacetate, where did we see that? Citric acid cycle. But citric acid cycle happens where? In here. So that oxaloacetate is not part of citric acid cycle. Okay? It is part of this shuttle. It is also part and can be used in something called gluconeogenesis, because oxaloacetate is part of that reaction and it happens in the cytosol. Okay? But check what happens. The NADH can be transferred to the oxaloacetate to make malate. Wait a minute. You learned a reaction, oxaloacetate and malate, right? But it was the opposite direction because it was in citric acid cycle. This is not citric acid cycle because it's happening in the cytosol. Now, what's important about malate is that it can go through a shuttle and be put into the mitochondria. Okay, what happens is an alpha-ketoglutarate has to come out as this goes in. That's okay because look at this. Alpha-ketoglutarate has to be part of the reaction for where we started with aspartate. So there's kind of a balance there. All right. The malate, now that's inside the matrix, gets converted into oxaloacetate. That is a citric acid cycle reaction, and I produce an NADH. Now check this out. NADH on the outside, NADH on the inside. Then a reaction occurs, okay, and what happens is aspartate comes out, but glutamate has to come in. Well, that's okay because glutamate has to be used here, and this alpha-ketoglutarate that's produced goes there. And wait, where do I get that glutamate? I get the glutamate here, so it's all kind of connected. Those 2 little molecules go back and forth and keep it all balanced. Here I produce NADH inside the matrix, and I can get the full 2.5 ATPs for each one of those, and I can get more ATP, 2 more ATP, if I use this shuttle. So obviously, I can get more energy. I use this shuttle all the time, yes? Why not? I should use this thing. Okay? Here's the problem. The malate aspartate shuttle is much more efficient, but the only way that that works is if there is more NADH on the outside of the mitochondria than on the inside. Very rarely does that ever happen. There are a couple tissues that do that. The heart is one of them that can allow that to occur. Almost every single other tissue that we have, the balance is not right. So I have to default back to the glycerol 3-phosphate shuttle. So when someone asks you how many ATPs you can create from one glucose, that's not enough information to answer the question. Okay? The maximum number you can create is 32. The most often created is 30. And it's because of the shuttling. And it is because that most often the NADHs from glycolysis are shuttled in a way that I lose some energy. And that's through the glycerol 3-phosphate shuttle. So it could be as many as 5. Very rarely does it happen. But it's almost all the time just 3. We produce 2 ATP in glycolysis itself. We produce from the pyruvate oxidation, okay, in the matrix, converting them from pyruvate to acetyl-CoA. I get 2 NADHs, which times 2.5, I get 5. I get 6 NADHs when I go through the citric acid cycle, okay, 2 times, because I have 2 acetyl-CoAs from glucose. I get 15 from that, 6 times 2.5. I make 2 FADH2s in 2 rounds of the citric acid cycle, so that's 3. And then I make 2 GTPs officially, but we count them as 2 ATPs, and so I can get 30 or 32. My daughter is taking human anatomy and physiology right now, the nursing course, and their textbook says you can make 38 or 36. And so you will see sometimes a debate, is it 36 or 38? The 36-38 number was when they calculated the proton flows differently. The more refined number now is 30-32. So if you look up on the internet, you will find the answer 30, 32, 36, and 38 for the number of ATPs that can be synthesized from one glucose. 38, 36, totally wrong. Okay? 32 the most, the maximum number, 30 the most often produced. Okay? That's important. Now, one other thing that happens in the production of ATP. If you take wood and you oxidize it, a hydrocarbon plus oxygen, you burn it. It creates energy. What is the energy that it creates? Heat. Yeah, that's where you take your marshmallow and you get your graham cracker and you get your chocolate and you make your s'more, right? For us, 34.7% of the energy was made into ATP, we calculated. Where does the other energy go? We don't lose energy. Remember, we can't create nor destroy energy. It actually is all those electron flows, the energy is released as heat. We consume food and we are warm-blooded because some of the energy that is lost as heat for us. And so this is how we maintain, one of the mechanisms we maintain body heat. Okay. All right, there's some special circumstances in which we can use this process only to create heat, or more effectively create heat than making ATP. Okay? And it's this way. What we do is we use our sugar and we break it down and we create a gradient, and the gradient creates ATP, right? And our ATP concentration really drives whether things are moving or not, because the concentration of ADP and ATP control a lot of the enzymes that we looked at in this process. All right. The molecule 2,4-dinitrophenol, DNP, all right, was found to do something that was kind of interesting. And it was initially used as a diet pill. Now, what did it do? Okay, when it's in the solution, in its ionic form, it's really, really polar. But what can happen is it can bind to the proton, become nonpolar, much more nonpolar, and then just diffuse across into the matrix. I break the gradient, but I haven't made ATP. So the concentrations in the cell say I still have high ADP, no ATP, so I continue to burn stuff. And it gets moved across the membrane, and what I can do is break the gradient. All of the energy is converted into heat, no energy is converted into ATP, and if I need to break down energy, I'll do sugar to a point, and then the body says, hey, I need my sugar from my brain, so I'm going to start breaking down proteins and fat. And so, ooh, great diet pill, right? I will take this pill, and I will make my body force it to break fat. Now, if you're trying to lose weight and you have a diet pill and you start losing a little bit of weight with one pill, what is the next step? Take a lot of pills to make a lot of weight loss. Okay, if you do a little bit of this, yes, you do break the gradient, you do convert it into heat, and you do break down fat. But if you do too much of this, what will you not produce? ATP. And if you don't produce ATP, you die. So it wasn't a very effective weight loss drug, okay? But it did mimic something that actually happened naturally.
Would it be effective in low doses?
It could, but do you want to walk into that organic lab, pull out the bottle of 2,4-dinitrophenol and take a scoop? There are other issues about it, and it binds to other places in the body, and so it was thought to be a wonderful. Some of the lab animal studies didn't go well. So, all right. This does mimic something that happens in natural processes. Instead of having the organic molecule break the gradient, there are tissue types, and it's called brown fat, that creates an extra protein called thermogen. And the brown fat, as opposed to the clear or milky fat that we have, it has a whole bunch of mitochondria. The whole point of that is those mitochondria and those fat cells do break the gradient by using the protein thermogen. Those fat cells do not create ATP at all. All they do is create heat. You find brown fat in both hibernating animals and newborns. Some of that baby fat. In hibernating animals, you need the heat to keep them warm while they're not eating anything, and their normal fat will produce the ATP they need. In babies, their weight is so small that their loss of heat is much higher, so they have to have this breakdown product to keep their heat up. And so the thermogen is an uncoupling protein that uncouples or breaks, that allows the gradient to flow through, and that's used up during hibernation, or you lose your baby fat over a period of time, but it keeps you heated. And so there's a natural thermogenic process that happens in certain adipose tissue. And so we can get heat from the oxidative phosphorylation process in a natural way. All right. we are at the end, and I have finished lecture material in the middle of convocation. So that means can't have an exam until when? Next Tuesday. So what we're going to do next time is come together. I'll review the exam. We'll go over what kinds of things that you need to be studying. I will let you know right now. You will, I think there's a 20-point question. Okay, so it's a biggie. You will have to reproduce either glycolysis, and that is the 10 structures of glycolysis, or the 8 structures of citric acid cycle, and draw them and name them. And you would have an arrow, and you'll have to label what comes in and what comes out for each of those reactions and the enzyme names. Okay? Now, I tell you this up front so that you can reproduce those structures and those names, and this one should be come in, get that done, and not think about that question, right? Just the time it takes to write those things down. If by chance you get to the exam and you have to think about that as you go through, that's not how I intend that time to be used, okay? Now, you have to know one of those two. I will choose which one. And so I've narrowed it down to 2. So, yes. All right. So, if nothing else between now and the next time we meet, at least that could be something that you study, because that's a big chunk. But 20% of the entire grade, I'm hoping for you, that's 20 points in your pocket, right? Because you should be able to get that done. That's straight up regurgitation of information. Yes? Just the structures, no mechanistic arrows on that at all. No mechanisms. Just the 10 structures or the 8 structures. Okay? And you can write ADP and ATP. I won't make you draw out ATP and won't make you draw out ADP. All right? You can do that? Yes. And the enzyme names also. Yeah, so from glucose to glucose 6-phosphate, hexokinase. Right? Okay? And so, drawn, yeah, like that at the top, and yeah, one of those two, yep, just like that. Okay? Now, I made you do the arrows as we are going through. I will tell you this, that one of the bonus questions that I have on the exam, I have 2 mechanisms that you could choose to attempt on the bonus. You don't have to do it. It's totally bonus. Okay? And so there are 6 points on a question, and there are 2 mechanism opportunities to earn points. But you don't, it's all bonus. You don't actually have to get it right to make an A, but you can, and you will have seen that type of logic in the reactions that we saw within the chapter that we covered. But there is no, you have to know a mechanism for the exam. It's just, if you got it, you get something better, okay?
The mechanisms from either glycolysis.
This could be any one step between one. I would draw what it is and what it's produced with anything that is needed, and then you draw those few steps. You know, it's not a long process. It's just 1-to-1, okay? It would be something like going from from glucose, which I would draw the whole thing out, to glucose 6-phosphate, I would draw the whole thing out, and I would draw out ATP for you. And then what you would do for that. And then you put the arrow, you put the mechanistic arrows. But that's all on the bonus.